AMALearning, you're about to. Specification point 3.6. Explain how DNA can be extracted from fruit. To successfully answer any GCSE questions linked to this topic, you will need to know the process through which DNA is extracted from the fruit, and also the reason why washing up liquid, salt, protease and ethanol are added. Extracting DNA from fruit is a practical demonstration of how DNA can be isolated and observed using simple materials. How can DNA be extracted from fruit? You will need the following materials. A fruit such as a kiwi or strawberry, because it has lots of cells. Additionally, salt, washing up liquid. A heat source, a filter, and chilled ethanol. You can also add protease. This is added to break down the proteins in the nuclear membrane. The nuclear membrane is the membrane surrounding the nucleus. It must be broken down to access the DNA. What is the process for extracting DNA? Step 1. Peel the skin off the fruit and mash it up. This breaks open the fruit cells. Step 2. Mix the fruit pulp with a solution of salt and washing up liquid. Salt helps to separate the DNA from proteins. Washing up liquid breaks down cell membranes, releasing the DNA. Step 3. Heat the mixture gently at 60 degrees Celsius for about 5 minutes. This denatures enzymes that could break down DNA and aids in further cell lysis. Step 4. Filter the mixture to remove solid fragments, leaving a clear filtrate containing the DNA. Step 5. Cool the filtrate using an ice bath, then pour chilled ethanol onto the top. DNA is insoluble in ethanol, so it precipitates out as a visible stringy white substance. This experiment shows that DNA, although microscopic, can be extracted and observed with the right tools. It's a great way to connect theoretical knowledge of DNA structure to hands-on application. Let's apply what we have learnt to a past paper style question. This GCSE style question asks us, explain why washing up liquid, protease and ethanol are added when extracting DNA from strawberries. The next part of the video will help us to answer the questions. Why are washing up liquid, salt, protease and ethanol added to the mixture when extracting DNA from fruit? Washing up liquid is added because it breaks down cell membranes, releasing the DNA. Salt helps to separate the DNA from proteins. Protease is added to break down the proteins in the nuclear membrane. The nuclear membrane is the membrane surrounding the nucleus. It must be broken down to access the DNA. Finally, DNA is insoluble in ethanol, so it precipitates out as a visible stringy white substance. Finally, let's see how much we remember from the previous part of the video. The first part of the question asked us to explain why washing up liquid was added to the mixture. And the answer, washing up liquid is added because it breaks down cell membranes, releasing the DNA. The second part of the question asked us to explain why salt was added to the mixture. And the answer, salt helps to separate the DNA from proteins. The third part of the question asked us to explain why the enzyme protease was added to the mixture. And the answer, protease is added to break down the proteins in the nuclear membrane. The nuclear membrane is the membrane surrounding the nucleus. It must be broken down to access the DNA. And finally, the fourth part of the question asked us to state why ice-cold ethanol was poured into the filtrate. And the answer, DNA is insoluble in ethanol, so it precipitates out as a visible stringy white substance. Our animations were designed by experienced UK-trained teachers. We have been achieving amongst the best results in the country for many years, and we decided it was time to share our success. Have you just AMALed and want to AMAL some more? Don't forget to like, subscribe and share to keep yourself up to date with the latest uploads. AMALearning, You just did.